good to be here. When you're being introduced like that, you start feeling that like you're very old. Yeah, I did my undergraduate at Tunza starting in 1980 to 84. That sounds like ancient to most of you, right? Yeah, but I'm still young at heart. So, okay, so what I thought I would do is basically just take you briefly through what the science behind vaccines are, what the science behind vaccines is, and how they work, how they are, or why they are important. And mostly because I think listening to Pastor Paul preach about love, vaccines have generated a lot of upheaval, especially in the evangelical circles among Christians and so on. People have been very hesitant to get vaccinated and things like that. It's like suddenly we kind of thinking like these are new things. Okay, how many of us are vaccinated in here? Okay. I hope you are not you are unvaccinated because you are hesitant. You are not sure about what is being injected into you and stuff like that. So hopefully, after I've gone through this, I'll maybe answer a few of the questions you may have. And if you have further questions, I'll be quite happy to continue the discussions after this. Now, I don't know how much time I have, please, or who is keeping track of the time so that if I... 25 minutes, okay. So that's my name. As was introduced, I teach in the biology department. I'm also the head of department at Trinity Western University. I teach mostly first year biology, the cellular components, and then I also teach microbiology to biology students as well as nursing students, and then I teach biochemistry as well. And vaccines is one of the topics that I cover. I cover this topic in about 2 or 3 lectures, and so I'm going to condense that into just about 20 minutes. Hopefully, you'll be able to get something from that. Okay, so some of the questions that I want us to consider as we look at vaccines. First, I just want to pose the question, how does the body respond to infections by a pathogen? Now, the body is encountering pathogens almost every minute, okay? And so the body has to respond to that, and then talk about what is immunity. What do we talk about when we talk about immunity? What are vaccinations? What is immunization? I'm sure these are terms that you've come across. Many times we come across terms that we think we know what they're talking about, but I think it's good to at least define what they are. And then I also want to just introduce you to the central dogma of biology. One of the I think in theology, we talk about dogmas, right? We also have dogmas in biology. What are some of the central truths in biology around which biology is built? And because of the central dogma, how then can we make vaccines based on some of these central dogmas? And I'm going to concentrate mostly on vaccines around the SARS coronavirus, the one which causes COVID-19. And asking the question, are these vaccines any different from other vaccines that we've encountered, which I'm sure from childhood you've been vaccinated against polio, against mumps, against measles, against all sorts of things. How different are these vaccines? And then how are vaccines tested before they are released for use in humans? Because a lot of times the question is, hey, are we being used as guinea pigs? And what is happening, and so on, okay? So that's where we are headed. I like starting, they talk about vaccines with this Psalm 139:14 to 17. The psalmist says, I praise you because I'm fearfully and wonderfully made. Your works are wonderful, I know that full well. My frame was not hidden from you when I was made in the secret place, when I was woven together in the depths of the earth. Your eyes saw my unformed body. All the days ordained for me were written in your book before one of them came to be. How precious to me are your thoughts, God. How vast is the sum of them. It's like the psalmist is just pondering about his body. Okay? I hope every one of us can say, I'm fearfully and wonderfully made. And as it comes to our fight against infectious diseases and pathogens, the body is actually fearfully and wonderfully made. God has put a lot of thought in your body, in my body, to encounter a lot of these things that we encounter, like pathogens. Okay? So I thought I would start with that. When you look at the body and how it fights against infectious diseases, there are two major ways in which the body responds to infectious diseases. You encounter a bacteria pathogen, okay? Or you encounter a viral infection, and so on. Two major ways in which the body is responding. Okay? The first one is using the innate immunity, as we call it, and the second way is to use the adaptive immunity. The adaptive immunity is also referred to as the acquired immunity or the specific immunity. Adaptive just means that the body is adapting as it encounters the pathogen. Okay? Some of the pathogens it encounters for the first time it has never encountered before. And so it has to start processing those pathogens in order to be able to respond to those pathogens. The innate immune system normally consists of things like the skin. All of us have skins, right? When a bacteria lands on your skin, it has to get into your body before it can start causing any diseases. Acidic environment. Do you know of any place in the body which is very highly acidic? The stomach. And so you swallow some bacteria, get into the stomach. Most of those bacteria are destroyed by stomach acids. Some of the pathogens are destroyed by stomach acids, but others are able to survive and proceed to the small intestines. The secretions, secretions in the eyes, the mouth, the saliva, all these are endowed with a lot of chemicals that are constantly dealing with pathogens as we breathe them in, as we swallow them, and so on. Okay? The mucous membrane. A mucous membrane lines the respiratory tract, it lines the urinary tract. All these areas trap pathogens so that they don't get into the body. Okay? The cilia, cilia in the respiratory tract, these are hair-like structures. So when you're breathing, pathogens land into the cilia moves the pathogens to the mouth, to the nose, so that you can sneeze, cough. All those are responses trying to get rid of pathogens, okay? Now, if a pathogen manages to kind of breach the first line of defense, we call this the first line of defense. So the pathogen now enters the body. The body has other ways in which it deals with that. This is still part of the innate system, which we are born with. Right from childhood, everyone is born with a skin, everyone is born with a mucous membrane, everyone is born with all these. We're also born with other cells like phagocytic cells. Does anybody know what phagocytes do? These are basically, yep.
They know bacteria.
Okay, these are cells that are hunting bacteria, they are hunting viruses, they encounter them, they take them in and destroy them, okay? They are natural killer cells, they're constantly killing other cells. Defensive proteins. These are proteins that the body produces, which are toxic to bacteria, toxic to some viruses. Inflammation. Okay, we've all had inflammation before. That's a response that the body has in order to try to deal with an infection. Okay? But of course, sometimes inflammation goes crazy, the body temperature goes too high, that becomes a problem. But normally, normal inflammation is just the response that the body has to try and deal with invasion by pathogens. So these two, the first line and the second line of defense, are part of the innate immune system, something that we are born with. As soon as we are born, we are all endowed with all these. God has given us the ability to ward off all these infections. The response of the innate immune system is very rapid. It recognizes a broad range of pathogens, almost anything that gets into the stomach, it can deal with, and so on. Mucous membrane is not choosing to say, okay, I can trap this pathogen, I won't trap this pathogen. It's dealing with everything the same way, except there is no memory. It's like each time the body encounters pathogens in this fashion, it's responding almost like it's beginning from the scratch. Okay? When these lines, the first two lines, are breached, you then get to the so-called adaptive immune response. Now we get to this response here. This tends to be slow. The response is much slower. And it tends to be very specific. And most of the times that's why we refer to this as specific immune responses. The other thing that is different about this response is that it tends to have a memory. When the body first encounters a pathogen using the adaptive immunity, it will remember that, oh, I encountered COVID-19 virus, or I encountered a measles virus 10 years ago. Okay? Now, why this is important is because the first time it encounters the pathogen, the body starts to process to try and figure out how to defend itself against that pathogen. Okay? And after it has processed that information and it keeps a record of that, the next time the body encounters the same pathogen, the response will be much faster and it will be more aggressive. Okay? So keep that in mind. So there is the first encounter where the body is encountering something. Say, for example, a baby first encounters, say, the flu virus. Okay? The baby may get very sick. Okay? Because it's the first time the baby is encountering that. The next time it encounters that, the chances are it's not going to get very sick because it has a memory. The body is remembering that it has seen this before, and therefore it mounts this secondary immune system, which is much faster, which is more aggressive as well. Okay, so that's why we tend to refer to this as specific or adaptive. It's of two types. There is the defense against pathogens that are in body fluids, things like the blood, for example, the lymphatic system, and so on. Anything that gets in there, it will be dealt with by antibodies. Okay? And then defense against pathogens that are inside the cell. When a pathogen gets into your body cells, it means that antibodies cannot get to those pathogens. And so the body then has to develop other means to deal with that. It has what is called cytotoxic T cells. These are cells that are hunting other cells, your normal cells, which have been infected, and it will destroy those cells. You see that? For example, when a virus infects your body, the virus on its own cannot multiply. It normally gets into the cell and then starts multiplying in the cell. So that cell which has been infected is now in trouble because it's become a factory for the production of viruses. What these cells will do is to look for those cells that have become viral factories and hunt them out and destroy them. Okay, so that's what the cytotoxic T cells are doing. So my concern will mostly be on the antibodies and how they're being used to protect us. Okay, so what are vaccines then? Vaccines are normally just preparations of antigens used to induce the protective immunity. And the protective immunity that I'm talking about is the adaptive immunity, the last line of defense that we're talking about. Okay? And what we're looking at is if this is a pathogen can be recognized by antibodies that have been produced against that pathogen. And once the antibodies bind to that, they start to lead to the destruction of the pathogen. Okay, these antibodies can be recognized or can recognize different aspects. So these are just pictures of the COVID, the SARS coronavirus, the cause of COVID. It has these structures on the surface. They're actually proteins. Antibodies will normally recognize those and bind to those in order to start the destruction of the COVID virus. We'll talk a little bit about that later on. So, when your body has responded to this infection and it has produced these antibodies that are able to recognize specific pathogens, we now talk about vaccination. The process of introducing a vaccine into the body. Now, this vaccine can be the pathogen itself, or it can be a bit of the pathogen that has been introduced into the body, so that the body can then start keeping track of what caused the infection in the first place. Okay? Now, I talked about the body encountering the pathogen for the first time. This is a measure of the concentration of antibodies in circulation when you first encounter the pathogen. Say, for example, you first encounter the SARS coronavirus. Okay? The body, this is the first time you get infected. It takes about 5 days for the body to process the infection, what is it that is causing the problem, and then it starts producing antibodies. Okay? And the antibody level goes up. Within about 2 weeks, antibodies will peak to about that point, and then it starts to go down. So you remember when we were talking about quarantine. How many days were you being told to quarantine for the first time?
6 months.
6 months. Anybody remember? 14 days. So we're expecting that by the 14th day, the body would have produced enough antibodies to deal, at least to manage the infection. Okay? Because that's the way the number of antibodies in the body are produced. Now, notice that the second time you encounter the same pathogen, can you see what's happening here? How many days is it taking for the level to increase? Here it took about 14 days to 15 days for the amount to be very high. Here it's taking roughly about 5 days. Now, can you see the amount that is produced? Okay, it's much higher, and the production is much quicker. So we're saying more aggressive, much faster. And so the taking care of the pathogen then tends to be much faster. Okay, so this is what we call the primary response, the first encounter, the secondary response, the second encounter, or any subsequent encounter with the same pathogen. Does that make sense?
Yes.
So, that's what the body is doing. And so when we're doing vaccinations, basically what we are doing is to introduce a piece of the pathogen so that we train the body to respond with a primary response. So that next time the real thing, or the body encounters the real thing, it produces the secondary response. It's more aggressive, able to deal with the problem much quicker. Okay? Now, the question is, when you are vaccinated, does it mean you'll never be infected again? No. The body has just been trained so that it can respond to the infection much faster. You see that? Okay. So keep this in mind when we look at the vaccines. So immunization is basically what we are saying when you're in this region here, then you are immunized, because now you can respond with a secondary response. Now, I have a second graph here where I said, okay, you will then encounter a second pathogen. So maybe here it was SARS coronavirus, here you encounter the influenza virus. The influenza virus is not going to produce the secondary response. It's going to go through the first primary response, and then upon subsequent encounter with the same influenza virus, it will then produce the secondary response. That's why we talk about this type of immunization as being specific, because immunization against one pathogen is not going to protect you against a second pathogen. Now, when we dealt with the coronavirus, we talked about the Delta variant. What are the other variants? Can you remember? The Omicron. Okay. Now, because it's the same virus, what the Delta virus did was to mutate, change a few of the characteristics on the surface, so that it's not exactly the same when we look at the Omicron virus. It has some changes. But there's still a few things that are similar between the Delta variant and the Omicron variant. So the vaccination you get against the Delta virus, you may be able to respond in the secondary response against the Omicron, but maybe not very, very well. Can you see that? Because there are a few changes that are taking place as the virus mutates. So how do these antibodies that we are talking about protect us? What the antibodies are basically doing, if this is a virus, the antibody will bind around the virus, so that the virus now is not able to get into the cells. Can you see that? If it's a bacteria, the same thing. The bacteria is not able to get into the cell to cause disease. Toxins, the same thing. Some toxins like the tetanus toxin. You also get immunization against tetanus, right? Basically, you're producing antibodies that can then recognize the tetanus toxin. Okay? Any questions so far? All right. So, the central dogma, how do we use this to prepare the vaccines that we're talking about? The central dogma in biology just says, in biology, each cell has its DNA. Replication will reproduce the DNA. You get injured, you cut yourself, the cells multiply and close up the wound. It's because they're producing the DNA of the cells that were injured so that they can produce the proteins that are required to close up the cell. The DNA, in a process of transcription, they produce RNA. So if you're looking at what is happening here, this is now for you scientists, you know what a cell looks like, right? It has a plasma membrane, it has a nucleus. In the nucleus is the DNA. The DNA is transcribed into RNA. The RNA is taken into the cytoplasm. The cytoplasm, that's where the proteins are being made from the RNA. That's basically the RNA being used to make proteins. Viruses, some viruses are DNA viruses. So they can go through this process. The only problem is that viruses don't have the proteins that are required to take the DNA into RNA into proteins. So when they infect the cell, they use the proteins from the cell to carry out this process. Okay? Some viruses are RNA viruses. They can also reproduce by reproducing the RNA so that the RNA can then be utilized to make proteins. So if you look at this, what has happened here, here is a virus that is infecting a cell, gets into the cell, it releases the genetic information here. That is used then to make more genetic material and the proteins that are required, and then the viruses are assembled in the cell, and eventually the viruses start exiting. And you see that? Now, this is a human cell that is infected, it's producing viruses, and this is where the cytotoxic T cells that I talked about come in. If the cytotoxic T cell recognizes that, it will then destroy that so as to stop the process of this cell being a factory of viruses. Okay, I don't think I need to talk about this. I'll just jump this. So how are vaccines then produced? Okay, now I want you to look at this. We are looking at viruses, vaccines against viruses, specifically the COVID vaccine. If this is your COVID virus, the COVID virus has specific proteins on the surface, it has some proteins inside, and then it has the RNA, which it uses to make itself, to reproduce itself. Okay? So, in the process of making vaccines, we can use various ways of making vaccines. The first one is the viral-based type of vaccine production, where you take this same virus in the lab, you treat it in such a way that it's no longer pathogenic, but it's still a virus. It's still the real thing. And then you inject it into the body. Okay? So the body can now recognize the real thing, but it's not pathogenic. Okay? So the body responds in the primary response so that when it encounters the real thing, which is pathogenic, it can go through the secondary response. So that's in the case of live attenuated. So this tends to be very close to the real thing, and vaccines that are produced using this tend to be very effective. And so you've heard of the polio vaccine. I think one of my nieces was going around, I think there is a campaign to revaccinate people against polio. Okay, the polio vaccine is a real thing, but it doesn't cause polio. So you put it on the tongue of babies and let them swallow that so that they start the process of the primary response. That's the Salk vaccine, okay? The chickenpox vaccine is the same way. Similarly, the measles vaccine is basically the same thing. You're taking the real thing, treat it in such a way that it's no longer causing problems, then you use it as a vaccine. But you can also take the real thing and kill it. Okay? So in this case, in the first case, these can actually multiply, but they don't cause disease. In this case, they are not able to multiply, but they carry some characteristics of the real thing, which the body can then use to recognize it. Okay? And so another polio vaccine, the Sabin polio vaccine, this is the one which is administered mostly in North America. Here we administer this mostly. The influenza vaccine, the rabies vaccine, the same thing. Okay? We can also use just a piece of the vaccine, the virus. So here is the virus. If we take a piece of that, here is a protein. If we inject the protein into the person will produce antibodies against this protein. But when they encounter the real thing, which has proteins on it, what do the antibodies do? They still recognize it and are able to take care of it. Okay? So we talk about this as subunit type. And then there is the viral particle type. I won't go into this. And then, of course, the way the antibodies are being used to do that. Now, this has tended to be a slow process. Okay? Let me now bring you back to the SARS coronavirus. We now talk about what we call the next generation type of vaccine production. In the first case, we needed to have the virus, the real virus, so that we can either kill it or use it to produce the attenuated type or purify some protein from it. And most of the times, the problem is that when you're doing that, that type of producing vaccines tend to be very long. And it also is a problem. If you're dealing with something that is very dangerous, not everyone can work with that. Okay? The labs that you're required to work with are very few. I don't think we have any labs that will work with, for example, the Ebola virus and things like that. Okay, so that becomes a problem. And that starts to leave just a few labs to work with the viruses. Now, in the case of the next generation sequences, here we don't need the actual virus. And to give you an example, the SARS coronavirus genome became available in January 2020. And by March 2020, vaccines had already been produced, which were undergoing clinical trials, just within 3 months. Most of the vaccines that we've been using have taken years to develop because they use the classical type of vaccine development, okay? In this case, we're just using genetic material, the DNA or RNA. We either take a piece of the DNA or a piece of the RNA, package it into some vector, and then introduce it into the body, allow the body to make some proteins, except these proteins are viral proteins. And the body responds to those as foreign particles, and they're able to recognize what these proteins are coming from. Okay, and there are various ways in which you do that. So it can be DNA, like in the case of the viral vectors, or they can be RNA particles. Now, the RNA particles is like the bare bones type of vaccine. It's the simplest vaccine that you can introduce in a body. Okay? DNA, more complicated. RNA, very simple. The only problem with the RNA vaccines is that although they are quick to manufacture and very effective, and you can ensure the purity of the RNA vaccines, RNA tends to be very unstable. And so most of the times you require very cold storage temperatures, minus 82, minus 70, and so on. So a lot of times the rollout of messenger RNA vaccines tends to be very slow, unless you have the facilities that are required to do that. Okay? So, but the process, the principle is still the same. You want to introduce this RNA into the body then uses that RNA to make a protein, except the protein that is being made is the viral protein, so that the body can then respond and produce antibodies against that. Okay. This is just an indication of the type of vaccines that people have tried to produce. This was from December 2020. Okay. They were gene-based, so DNA, RNA, so many of them are under development. Protein-based, those that are taking a protein of the COVID virus and trying to make vaccines against that. And then, of course, the virus types, not too many, and the others as well. Okay? So many vaccines have been tried, or at least are being made against COVID. But, of course, we've heard so much about the RNA vaccine. Okay, this is just the table to show you some of the vaccines based on that table. The heat-killed whole virus, I think the Sinovac, I think is available in Zambia. That's the Chinese one. Storage temperatures 2 to 8. Then we have the purified components. There are others. I don't think any of them have been approved in Zambia. And then we have the non-replicating viral vector carrying pathogens. The AstraZeneca, I think this was one of the very early type of vaccines that were introduced, approved in many countries, and I think it's been approved here as well. And then you have the Janssen virus. It's also non-replicating vector type, approved in many countries as well. And then you have the messenger RNA vaccine. Okay, so there are many different types of vaccines that are being used, and many ways in which they are being applied to people. Okay, so just the big picture. I think I'll stop with this. When we look at vaccine-preventable diseases, this is just an example of polio in 1988, worldwide prevalence of polio. Can you see that? Almost all countries were affected. With aggressive vaccinations, there are only three countries, is it three or four? Three countries where polio is still endemic. And that's Nigeria, Pakistan, and Afghanistan. Okay, we know something about Afghanistan. There have been wars and so on. Pakistan, again, the same issue. Nigeria, the other issues with vaccine acceptance and so on. Now, because there are still these hotspots, these are viruses, okay? It means that somebody moving from Nigeria to Zambia who is infected will bring the polio virus here and will start the reinfection process again. So you cannot kind of keep your guard down and say, oh, there is no polio anymore, as long as there are countries where there are polio infections. Okay, just like the coronavirus itself. Coronavirus is still happening, right? Okay, so you cannot say, oh, because not too many people are getting sick, therefore we've eradicated that, and let's stop the vaccinations. No, we can't do that. We have to keep going, keep doing that. Okay, I think I've used up too much time. I'll stop here and see if there are any questions.